thank you to the Connection team, Thomas and Zan and the rest of the team for just another fabulous day. I absolutely, I know we started with, with laying out the concept of metabesity and defining it yet again. And I'll add something to it. What Metabesity is frankly the best conference of the year. This is just a tremendous and exciting and just a big tent for learning so many things. And this collection of people, and I realize a lot of us, certainly yours truly, miss the physical world, but we can do things in the virtual world that frankly we could never have done in the physical world, and that's going to give us maybe a nice problem to have as we emerge from the pandemic. So the purpose of this panel is, and I want to be clear that this panel is not, it is to allow some of the emerging companies in this space, and I think some really exciting companies, to tell a little bit of their story. But what I really want them to do is set their story, once spending a couple of minutes, setting out their story, and I charge them to do this in just a few minutes with a few slides, just so you can get situated and you can get to know the company and you'll understand the perspective that the presenter may be speaking from. But I think what all of these companies have in common is that they are participating in what is yet an emerging field. And, and I think this is, we all have this in common. I think what these companies also, I was struck by Aubrey's comment earlier today, which I don't know if some of you heard it, you may have been a little bit jealous. But what Aubrey said is that he's seeing essentially a windfall of money coming into very, very early stage programs, programs that are probably to some degree academic and maybe not even really fully translated. Well, the companies on this panel are post-translational. They are, for the most part, pretty early. They represent different status in terms of their financing, ranging from public companies to seed and in pre-series A. But what they share in common is really fascinating platforms. And I would actually think that despite the platforms that may be different, they share similar challenges. We're in a novel field. Many of these challenges, and this will bring this home a lot of what was discussed today, many of these challenges are challenges inherent in a novel field. The fact that we have ill-defined regulatory pathways, that we have massive considerations in terms of identifying patient populations, that we have long ways to go in terms of development of biomarkers, but I'm sure we'll hear some of that from Alex today. And that, and we heard a lot during the day about that. It's just fascinating. So the field is moving forward, but what this field really, really needs, in addition to more elucidation of the science and more clinical data, what this field really needs is a couple of successful companies and a couple of big exits. And I'm just going to be absolutely blunt about it, because when Aubrey talks about all the money flowing into the field, I see caution signs. Okay, I see signs that say, okay, but let's be very, very careful of what we wish for, because if we can't turn this into exits for investors, then the capital will not continue to chase the science, in this field. And that in fact is something what we want, okay? We don't want science chasing capital. We want for this field to advance. We want the capital chasing the science. We want this, to catalyze the field. So with that is my introduction. I think I'll just call on people in sort of a left to right way in the way they sit on my screen. The chat is open out. Send your questions in as people requested before via the Q&A, but I'll also monitor the chat. So we'll keep questions going. Ask them at any time. If you do ask a question, please tell me which presenter you want to direct it to, or whether it's a general question for the panel. So on my screen in the upper left is Steve Engle from Cohbar. So I'm gonna call on Steve to kick this off.
Thanks, Ed. Can we pull up the slides? So just briefly, Cohbar is focused on turning mitochondrial sequences for peptides into novel therapeutics. Next slide. So you've heard that mitochondrial dysfunction is one of the hallmarks of aging, and that's because it underlies many of the diseases. Next slide. So on your right is probably at least $500 billion worth of inpatient payments. Mitochondrial dysfunction affects about 5 out of the 8 leading causes of death. If you solve the problem, because there is this underlying effect, you're likely to have positive effects on several diseases at the same time. Next slide. So the key discovery by Cohbar's founders, including Nir Barzilai, who you've heard about at the conference already, and Dr. Hassi Cohen, who made the original discovery, said peptide sequences are encoded in the mitochondrial genome. This is a shocker. Nobody expected it. The mitochondrial genome is separate from cellular genome, and so it's a big surprise because it's very small, relatively speaking. The second big surprise was that peptides regulate multiple organs and systems in the body. So far beyond being the powerhouse of the cell, mitochondria is involved in sending out peptides that are communicating to multiple organs. And researchers have published studies of peptides, metabolism, oncology, and longevity. And what we have at Cohbar is a technology platform. We have over 100 peptides and over 1,000 analogs that we've discovered. And what we're doing is basically imagine them up on the wall, and we're pulling them down one at a time, turning them in the lock to see what they do. Next slide. So these are the 5 programs we have going, 1 in the clinic, and then 4 others. So you get a sense by reading out the indications. There's quite a diversity here. It's also represents, each one of these represents a different program with different molecules, and each one has multiple indications. So it's a pretty exciting time. Ed asked us to talk about that. As you can guess, having 100 sequences already identified puts us in a very interesting position of essentially just going through these and finding out as quickly as we can what they do. And this gives you a little bit of the sense of the breadth between oncology and working in the area of ARDS as well as in the pulmonary area. And of course, the lead compounds in NASH. In terms of challenges, Ed asked, talked briefly about that. I think he's alluded to the problems with working in diseases where the clinical study designs are not clear yet, and where the regulatory pathways have not been defined. So in the case of NASH, certainly it's an area where there have been bumps along the road this last 2 years. For other companies, we feel very fortunate because our compound has a completely different mechanism of action, and we are indeed learning from everyone else. Thank you. Ed?
Let's come back to the, okay. Great. So I'll come back to the screen. Thank you, Steve. So again, I just want to remind you, and Steve, thank you for adhering to the format. I appreciate it, because I do want to have a lot of time, for discussion afterwards. And just to be fair to the presenters, understand that I really did challenge them for the executive summary. I mean, the highest level, basic, what business are you in? What's the pipeline? What's the promise? And what's the challenge? And I would like each presenter do that, like Steve, talk about what the promise is, and I think that was clear in Steve's presentation, and what some of the challenges are, and I want everybody to do that right afterwards, because I think, we won't then disassociate the challenges from the specific companies, but I am actually banking on the fact that by the time we're done, we'll find out that despite the fact that we have a diversity of different companies with approaches, we sort of have similar challenges, which are the challenges that characterize the field. And in terms of this meeting, this Metabesity and the challenges moving this field forward, I think that'll be a very helpful dialogue. So with that, I'm going to turn it over to Hanadie.
Great, thank you, Ed, and I will go ahead and screen share myself. Great, so just quickly want to say thank you so much for this invitation to participate in this really interesting, provocative conference on ending age-related diseases and really targeting metabesity. And so just to share a little bit about what we're doing at Juvena Therapeutics, it is our goal, and can you guys see my screen? Is that working?
We can, I can.
Okay, great. So at Juvena, we are dedicated to developing protein therapeutics that can promote tissue regeneration in order to cure disease and rejuvenate lives. And we ultimately want to treat a host of age-related and degenerative diseases. And the way we're really rapidly bringing our protein therapeutics to market is by taking advantage of state-of-the-art proteomics and machine learning, which I'm about to tell you a little bit more about. The secret sauce that we're using is that we are unlocking the unlimited regenerative potential of proteins that are secreted and produced by human embryonic stem cells, human pluripotent stem cells, that have the capacity to develop every tissue in the body, an entire human being. And what my colleagues and I first showed during my time as a graduate student at UC Berkeley and then extensively at Juvena Therapeutics is that by isolating a subfraction of the proteins that HECs produce and secrete, purifying and concentrating them, and then adding them onto aged human precursors and various different human cell types and in vitro disease models, we could witness this enhanced regenerative effect. So for example, on muscle, it enhances aged human muscle precursor function and myogenesis. In vivo, in an acute injury model, we see enhanced muscle regeneration, a reduction of fibrosis. When we add this cocktail of proteins onto aged human chondrocytes isolated from osteoarthritic knees, we see this improvement and enhancement of proliferation as well as survival. It can also promote survival of differentiated human cortical neurons in the face of A beta. And specifically at Juvena, we are first translating these proteins to the clinic for muscle regeneration and muscle diseases. And please ask me during the Q&A why we chose to focus on muscle first. And the way that we are at Juvena translating this cocktail of proteins to the clinic is by having established a very efficient identification, screening, and preclinical development platform, where we first, using state-of-the-art, unbiased, quantitative mass spec-based proteomics, really identified and quantified what the proteins in this subfraction of active molecules are. For example, these proteins produced by HECs have regenerative capacity, while the same subfraction produced and secreted by differentiated cells do not. And so it turns out that there are about 1,000 bioactive proteins in this complex mixture. And then we built a machine learning enhanced identification, high throughput screening, and extensive preclinical validation platform in order to identify proteins in the mixture that are driving the rejuvenation process for a particular tissue type. And upon identifying them and validating them in animal models, then extensively performed protein engineering and lead optimization to really turn them into active novel biologics that can be developed for tissue-specific degenerative diseases. Just to share a tiny bit about our machine learning toolkit that is growing and really enables us to rapidly identify and develop these proteins into therapeutics. We, for example, have several deep learning-based models that we've developed. We have a model that allows us to have better and more rapid actually identification and rank ordering of the proteins in our cocktail for their ability to enhance specific tissue types function and health. And that's done really through combining quantitative structure analysis relationships with natural language processing and the vast amount of scientific literature and information we have on structure function relationships to really predict what proteins might be working for a given disease. We then have deep learning based models that allow us to, for example, take bright field images of primary human precursor cells in these in vitro disease models and predict what their fluorescent labels and hallmarks of aging are and even relatively their biological age. And we've developed tools to help us enable us to rapidly quantify, for example, histology of injured tissues post-treatment with our therapeutic candidate. For example, regeneration, fibrosis, And ultimately, we're really developing tools that combine computer vision with proteomics and gene expression in order to enhance our predictive accuracy and really understanding of how these proteins individually and simple combinations may work and be turned into therapeutics for degenerative diseases. As mentioned, we have been focused with laser-like focus first on enhancing muscle regeneration. And we now have 3 protein therapeutic candidates in our pipeline that have been tailored to specific myopathies and degenerative muscle disease based on how they're performing, not just in, of course, in our high throughput screening, but extensively in specific preclinical animal models that represent the disease, as well as the unique signaling pathways and mechanism of action of these proteins, with our top lead, for example, treating dystrophies, sarcopenia, atrophy, and post-operative care. Ultimately, we'd like to really take advantage of this regenerative secretome and systematic approach that we've developed at Juvena to translate our regenerative library through our JuveNat machine learning enhanced platform into protein-based therapeutics that can really be used to treat a variety of degenerative and tissue-specific diseases. With that, I will stop there. Of course, share more. I look forward to sharing much more detail on Thursday. And just in terms of what I'm most excited about is really just, I cannot wait for us to really bring our first protein therapeutics to market to promote muscle regeneration in order to restore function and strength, because I truly believe that that will enhance health span and actually end up enhancing the function and health of multiple organ systems that are connected to muscle and really allow us to have many more years of disease-free, healthy aging. And as many of us can relate at this conference and panel, I think the biggest challenge really is regulatory at this stage, and happy to talk more about that during the Q&A.
Okay, thank you, Hanadie. We'll definitely get there. We'll get to the regulatory. I actually think I'm gonna, I'm gonna break the order a little bit because Alex is going to represent a very different approach. And again, we've had, Thomas and I had some interesting back and forth, which I know you were copied on, Alex. So I'm going to actually have you go last. And I'm going to keep the therapeutic pipeline companies as pipeline companies right now grouped together. So I'm going to call on Ben and then Ashish, and then we'll go to Alex. I just want to reiterate, I'm getting some notes because Thomas wants us to have a lot of good conversations. So let's try to be as crisp as we can on the on those brief presentations. So I'll turn it over to you, Ben.
Awesome. Thanks so much, Ed. Well, just to get it going, I'd like to thank Thomas and Zan and the Connexum team for pulling us all together here, and particularly for all the support that they've provided to Pano since we met at the Metabesity 2019 event. So this is our second Metabesity conference, and to say that these have been significant and supportive in the history of Pano would be a great understatement. We're really grateful to have the support of this community. So as the business guy on Team Pano, I'm going to do my best to run through these slides quickly to describe Pano's domain in broad sweeping concepts that really only a non-scientist can get away with. And I encourage the audience here to join the first session tomorrow morning, in which Pano's scientific founder will get into the biophysical weeds of mitochondrial biogenics with Doug Wallace and a solid panel of scientists in the field. So I'm going to sidestep deep technical discussion here, per Ed's suggestion, focused mostly on the business stuff. So interpreting our tagline on the cover page here, Pano is in the business of clinical development of patented small molecule drugs targeting mitochondrial ion channels. So we look at this as being the most mature available business model based on Pano's core science, if you will. Small molecule compounds, ion channels, not particularly innovative. Mitochondrial domain, the mitochondrial ion channels being a very elusive environment. And this, we believe, is our core sustainable competitive differentiation, our deep moat on what we're going to be building around. Next slide, please. Oh, great, thank you. So this slide explains Pano's alignment really with Connexum's Metabesity theme and puts this in the context of mitochondrial biology on the left side and the story of metformin on the right-hand side. So looking first at the left side, we've borrowed this figure from a 2011 Doug Wallace paper. Professor Wallace is one of the collaborators on the core discovery around which Pano is built. And the hypothesis described in this paper suggests that there's a mitochondrial dysfunction at the root of complex diseases of aging and even the aging phenotype itself. On the right side, we really see the inverse in the observation of the pharmacology and efficacy pleiotropic benefits of metformin. We see that metformin is a mild and transient inhibitor of mitochondrial complex 1, among various other proposed mechanisms of action, and seems to act in a manner that unwinds these various dynamics of dysregulated mitochondria on the left-hand side. So we really live in a world trying to align these as a result of our core discovery. Metformin presents as a pleiotropic drug, first line of defense for type 2 diabetes, a myriad other applications and clinical trials ongoing, despite some significant deficiencies of the drug. So we know that it's wildly useful. It's one of the most prescribed drugs in the world, particularly safe. That said, it's got low potency on its mitochondrial target, has poor access. It requires the OCT1 transporter to get around and to get into the cells. It's got an unresolved specific mitochondrial mechanism of action. Of course, it's impaired from a business model perspective as an unpatented drug. So we believe that there's an opportunity as we figure out what metformin is really doing within the mitochondria to develop enhanced pleiotropic drugs targeting the underlying mitochondrial dysfunction for this constellation of diseases that appears as effectively epiphenomena of mitochondrial dysfunction. Next slide, please. So drilling down into mitochondrial ion channels and the core discovery around which Pano is being built. On the left side, our working scientific and business hypothesis is that mitochondrial ion channels are very, very significant. They're involved in energy metabolism and all of the other beautiful orchestration that goes into making complex biology work, and mitochondrial channel dysfunction manifests in chronic complex diseases. So we're trying to bring it down to this biophysical, biochemical layer. Francesca Fiini, Pano's scientific founder in 2017 at UCSF, discovered a novel potassium ion channel associated with complex 1 that is inhibited by metformin. So we believe that this presents the central mechanism of action of metformin in the mitochondria. And our working hypothesis is that this is the root target of metformin that holds together the metabesity theme, the mitochondrial etiology of complex disease, the relevance of mitochondrial ion channels as pleiotropic drug targets, and particularly the significance of this potassium channel as a very important drug target itself. Next slide. So 3 pillars of our development strategy. On the left side, we're building out a mitochondrial ion channel platform. This is traditionally a very technical, manual, low throughput domain, and we're in the process of preparing to prototype moving that to a higher throughput environment. We have 2 drug development programs. One is based on the far right side on NMEs that we've developed that we believe are going to have greater access and potency on this potassium channel target. We're going to be developing them initially for diseases characterized by acute administration due to the unknown safety profile of the NMEs. And we're about to launch in vivo for colorectal and pancreatic cancer. In the middle column here, we've got a natural compound formulation drug development program where we've innovated a natural compound formulation based on 2 synergistic APIs, one of which has a known complex 1 target that's mild and transient, we believe works on the same target. It's a counterintuitive combination of these 2 APIs that gives us the ability to have a composition of matter patent foundation that sits on the bottom. So we have similar patent protection to NMEs. We benefit from the abridged 505(b)(2) pathway with the FDA. So we're developing these compounds, or this formulation rather, initially for diseases characterized by chronic administration. We're currently in the process of selecting our first cardiometabolic indication, and we're planning to launch in vivo program on colorectal cancer in parallel here in the next month or so. Next slide, please. So really in the business domain, when Francesca and I got together a year and a half ago, there was a lot of talk around our natural compound program as being grass and maybe quite interesting for, nutraceutical development, and all the talk about the TAME trial and the possibility of developing, anti-aging longevity drugs. And after much head scratching and really thinking about strategic precedents and all the carcasses by the side of the road in the consumer business generally, and the challenges of making claims and really getting access to capital and business model dynamics that are much more speculative in that regard, we decided to use plain old clinical drug development model. We believe that this is enabled by the composition of matter patent claim for the natural compound formulation that we have sitting at the foundation of this. So we're just finishing off our first animal models right now that are going to give us some data from which to choose our initial therapeutic indication here. And our objective is to find something that's capital efficient and risk efficient, get into phase 2 after a B2 IND in Q2 2021, and then using this Amarin model, really do sort of crosswalking and label expansion to get to the biggest possible TAM with a pleiotropic formulation that looks like it could be good for all manner of metabolic symptoms. So with that, I turn back to you.
Okay, thank you. Can you hear me? I don't know if I'm on mute or not. No, good. Okay, so thank you, Ben, and we are remarkably staying right on time. We've got about, we can finish the openings by 5:45. We'll have a good 15 minutes for discussion of some of these issues that have been raised. I've been noting a few of them, quite a few of them, and Ben, you didn't identify them all that specifically, but I saw them in some of your slides, so I will, I'll come to that, which is great. So why don't we go to Ashish?
Let me share my slides here. Okay, great. So, I hope to bring a sort of a somewhat orthogonal perspective to this discussion on multiple fronts, not just around the mechanism of action that we are targeting, but the class of therapeutics that we are developing in Glyscend, which lend themselves to many interesting opportunities, but also challenges from a regulatory and a clinical and market positioning perspective. And just to kick off this discussion, as a gastroenterologist, I very strongly ascribe to this notion that all disease begins in the gut, right? But this is lot more relevant when it comes to metabolic regulation. Many years back, I was fascinated when we learned that a common kind of gastric bypass surgery, which is the Roux-en-Y gastric bypass, where the intestines are rerouted and the stomach is shrunk in size, is not just effective for weight loss, but in 80 to 90% of type 2 diabetic patients, it almost cures, quote unquote, cures type 2 diabetes within 24 to 48 hours without these patients having lost an ounce of weight. And this was a fascinating insight, and for a long time, people didn't recognize or realize or understand why this was the case. But fast forward through a series of scientific investigations and device-based targeted approaches, for example, a endoscopic plastic sleeve that is placed in the duodenum, which is the first portion of the small intestine, we have started to appreciate and recognize the role of the small intestine, which is right after the stomach, and the first portion of the small intestine is instrumental in metabolic regulation. And it goes beyond just glycemic improvement, right? In the near term, these patients start losing weight, but in the long run, they also have significant insulin responsiveness. They lower their insulin resistance, but also the early data has started to show that it affects the macro and microvascular complications, including has a beta cell rescue property. But also now there is a big push towards glucose offloading in these patients. And the mechanism of what I call as duodenal exclusion, excluding the first portion of the small intestine, sits right center for facilitating this mechanism. So many years back, we took upon this challenge that could we recreate some of the effects of gastric bypass surgery, but with a pill, knowing how invasive, expensive, and burdensome gastric bypass surgery could be. And through many years of experimentation and research, we eventually came up with a material-based approach where we have now designed and synthesized materials through a robust preclinical candidate discovery platform, where now we have a material which we are planning to take into clinic next year. And essentially the way it works is the patients swallow a pill preprandially, and the material is released in the stomach, but is not active until it reaches the small intestine. And based on the pH activation and certain properties, the local luminal properties in the duodenum, this material is activated and is essentially complexes with the mucus. All of us understand that mucus is throughout the GI tract, lines the epithelium, which is a natural barrier, but in many ways this barrier has holes. And what we are doing is, through a material-based approach, is enhancing, complexing the mucus and creating a barrier and strengthening the barrier properties. And one of the beauties about our approach is that the materials that we have developed are entirely non-absorbed and have a short half-life, in that they're eliminated from the GI tract within 24 hours. They are not metabolized in the GI tract, and it essentially has a sort of a mechanical activation and mechanical effect, interfering with the neurohormonal signaling that happens through the duodenum, pancreas, and the liver. And this is our sort of a preclinical candidate discovery platform. But looking at in hindsight, what we have used here is we have utilized a lot of design principles, not just from the drug development world, but also the device development world, and have tried to marry them together, and then essentially invented our own sort of a polymer discovery platform. And front and center all along the discovery process was the regulatory and the commercial aspects of having safety as paramount, but also cost of goods, manufacturing, scalability, and how this would actually be administered in the clinic. And so this is a perfect confluence of decades of understanding of the gut physiology and its impact on metabolic regulation, with a sort of a novel class of polymeric compounds, although it's not as novel as one might think, because polymers have been used in the GI tract, and some of the sort of very successful billion-dollar drugs like Renagel are built using a polymer class of compounds. But what this does is provides us leverage around some of the regulatory and market aspects of how we position a therapy like this to target a new mechanism of action to treat an old but still a epidemic that still remains to be unsolved, which is type 2 diabetes. So with that, I hand it back to you, Ed.
Great. So thank you so much, Ashish. And Alex, I'm going to prevail upon you, and I know you have a series of slides. I really want to, and it's not fair to you, but I've cast you in the last role here, because we also have some questions that have come in from the audience. So if you can do this in a flash of 6 or 7 minutes, that'd be great. It'll give us time for discussion. Thanks.
6 or 7, perhaps. We will see. So, do you see my screen?
We do.
All right. So, hi everybody. My name is Alex Travanka. For those of you who don't know me, I'm calling in from Hong Kong. So it's almost 6 a.m. here. We are equal opportunity 7-day-a-week workers. So I wear 2 hats. One is the CEO of Intellico Medicine. It's focusing on end-to-end drug discovery. so artificial intelligence for end-to-end drug discovery. And another company which we successfully spun off out of in silico and already sold to a publicly traded company in Hong Kong. It's called Deep Longevity. It's focusing on deep biomarkers of aging and longevity medicine. So you can add me on WeChat here if you know how. Quick disclaimer, so Deep Longevity has been acquired by Region Pacific, Hong Kong's ticker symbol 0575, not investment advice, no forward-looking statements, etc. And the technology that we are embracing and pretty much ubiquitously using and developing is called deep generative reinforcement learning. So you've probably heard about deepfakes, the ability to synthesize really good high quality human facial data with specific properties that you want to have in those faces. Reinforcement learning is essentially AI strategy. So think of AlphaGo outperforming the best human Go players by learning strategy. So we combine those two and we essentially synthesize new data with specific desired objectives that we also learn at the same time. So we use this technology at in silico for biomarker discovery, target discovery, molecular screening, molecular generation, so that's where we're quite famous, clinical trial analysis. And at Deep Longevity, we focus on biological age prediction and monitoring using multiple data types, also biomarker development, and real-world data analysis. And now we even submitted a paper on mental health and behavioral modification. So a few words on in silico medicine. I'm going to be speaking in a day in the emerging companies showcase on both companies. So in silico is focusing on accelerating pretty much every phase of the pharma R&D. You probably know the numbers, but just to recap, we develop AI solutions that allow you to either run a marathon or do a sprint within any area. So either target discovery or target to hit or hit to lead. We have published some of those proof of concept that we did, a couple of years ago, where for a given target we generated novel small molecules that do not exist in known chemical space, synthesized, put them in vitro, validated in vivo. All the way in 46 days, so under 2 months. That was pretty cool. We have many of those cases, and I'm not sure why you think that we are not developing our own programs. We have 7 programs in development. Some of them are for novel targets that we've discovered using different data types. So one of the really cool programs that I can talk about just briefly is a program for a panfibrotic target. So far worked really nicely in preclinical NASH models. Several of them also in preclinical models of IPF in mice. We identified novel targets, generated novel molecules, and validated in vitro and vivo. So preparing for IND enabling now. And we also provide part of those solutions to big pharma. So if you go to insilico.com/platform, you see our platforms, and right now I think top 10 out of the top 20 big pharmas are standing in line for Chemistry 42 deployments. And Pandomics is going to be actually re-released on November 5th because we had to close it due to substantial demand. So paying customers are now using it. And our idea is to disrupt, digitize, and democratize. Again, we provide software, but at the same time, we develop our own programs. We're based globally, so raised 52 million to date from top biotech investors. But I really like to focus on aging. That's why I live. That's what we do. That's why we exist. And our idea is to disrupt traditional preventative medicine, where the doctor sees you only in the course of your recent maybe 10, 15 years in your age range. And doesn't look at you at the doesn't have a holistic picture of you in the course of your life. So we believe that it's possible to start developing the new field, longevity medicine, so AI-powered preventative medicine. And instead of trying to theorize whether aging is a disease, we put AI to work and check how the minute changes in the various features transpire into and transform into diseases. So basically, how do the hallmarks of aging become diseases? And nowadays, you can do that by first training on to predict age using multiple data types. We were, I think, the first groups to publish in this in 2016, using deep neural networks to predict age. Pretty popular paper. And then we started doing predictions of age and health status at the same time, looking at the differential features for age and disease. That's how we found our panfibrotic target. You can read about some of our work in this paper, the biomarkers of aging and longevity. And we recently partnered with Human Longevity on research. We are the high-level customers get our aging clocks, and we do a lot of research together. I think Human Longevity is one of the best and the most granular and very deep preventative medicine and longevity medicine enterprises. So in addition to this data, you can get aging clocks. And we've developed some really cool aging clocks using deep learning and using other technologies. Some of them are patented. We recently published a really cool deep microbiome aging clock together with Vladimir Gladyshev, one of the scientists who I deeply respect out of Harvard, really pioneer in the field. And you can read about our microbiomic aging clocks. It's actually a pretty good representative paper on how those clocks should be built. The workhorse clock is deep hematological aging clock. This is very simple blood tests. Those clocks are pretty responsive to interventions. We also do intervention-specific aging clocks now, and we create Age Metric. It's a system which integrates many of those aging clocks. We are pretty much data agnostics, and you can use it as a web platform. You can use it as an app. As a matter of fact, we just released it last week, so you might want to wait for another few days before we release the next version, because we found some additional issues that we want to fix and a couple features that we want to add. But basically, this week it's already available in the App Store, and you can play around with it. We're currently partnering with several pharma companies to do clinical trial, to assist with clinical trials using those aging clocks on some of the very simple data types that are very easy to collect. So thank you very much. Yeah, we have more than 130 scientists worldwide, and you can see us at Young AI or add me on WeChat. Very happy to collaborate. We're collaborating with many of the companies in the longevity space because that's what we live for. I think that aging research is the most important area of human development. That's why we like to partner with aging companies.
Thank you, Alex. And although it's 6 a.m. in Hong Kong, don't feel badly for Alex, because it's reported to me that he never sleeps. So so anyway, I appreciate you running through. It was a lot of material, and thank you for doing it so well. We do have, and by the way, Alex, just as point of correction, I'm aware that you actually have a pipeline. What I was trying to refer to is the breadth of your company, as opposed to the more focused companies, or breadth of your company or companies as opposed to the more focused companies we have here. So I'm glad we saved you for last. So I want to just, I basically have a question. I'm going to try to figure out if we can, if we possibly can, I've gotten permission to go to 6:05. So we can be 5 minutes late for virtual cocktails, and I hope that's okay. Because I do have some questions from the, they've come in from the audience. And I also have, some I've heard some things, okay? I've heard just in the more recent presentation from Alex, I heard the excitement, about the field. I heard some success, some exits, which I said earlier in my introduction is very, very important, and congratulations on that. I think that pushes the field forward, and we need more of these. But, I also hear challenges of the regulatory challenge. Okay, I think that we kind of all feel it and see it. Although some of our companies, and certainly Ashish, your company is working in probably a more conventional path. Certainly, Steve, you are in NASH. But there are challenging, settings too. Obesity is a challenging setting, let's face it. Ben, I think there are some interesting business model challenges, and I think you look like you might still be in search of that all important lead indication, right? So I think in terms of challenges, I think you presented it, incredibly well. So what I'd like to do is just jump a little bit to the Q&A. And actually, it's quite fascinating because you have a very, very compliant audience. So Hanadie, actually, because you requested to be asked why focus on muscle first, that's exactly what several people did.
Great. Thank you for the question.
First question to you, because you requested it and you got it. Why is Juvena focusing on muscle first?
Sure. Well, we're extremely interested in muscle because it is one of the largest internal organs. And as, for example, Tom Rando so eloquently described earlier today, it's highly regenerative. We understand many of the mechanisms underlying why muscle declines in function with age, and it is incredibly integrated into multiple other organ systems. We know that your muscle strength is highly correlated with all-cause mortality. We know that improvement in muscle strength and exercise can enhance cognitive function. It can reduce inflammation. Muscle is critical for metabolic regulation, for regulating heat, for blood flow, lymphatic system. And in many instances of disease, whether you are in the intensive care unit and within a week you're seeing 20% loss of muscle strength, it's so sensitive to environmental stimuli. Decline of muscle function and muscle wasting is a major side effect in diabetes, obesity, cancer, cachexias. And so we believe that by developing a protein therapeutic that can enhance muscle function, muscle strength, we will in fact use that and things like measuring the 6-minute walking test and physical battery of tests to in reality really be measuring your overall health span. And if we can improve muscle regeneration, prevent that atrophy in acute settings, enhance regeneration overall with age, prevent that degenerative process, I believe that then we'll see multiple other organ systems benefiting and overall a reduction in chronic illnesses that come with age and ideally this increase in health span.
Great. And although you didn't request this question, since you've got the floor, I'll just ask the second question that came into you. It's the last one, is why focus on proteins rather than exosomes?
We are focusing on proteins because that's where we see the regenerative effect, right? We specifically isolated proteins, which many others have also seen. For example, it's proteins in plasma that are in young blood are really driving the pro-health span rejuvenating effects that we see, this enhancement of stem cell function. Also, what's beautiful about proteins as the natural communicating messengers in the body is that they have historically been incredibly lucrative as a category of drugs, right? Recombinant DNA technology is in many ways the bread and butter of major pharma companies. And by developing a pipeline of biologics, I think this could be a very potent and rapid way to really translate these regenerative effects we're seeing into the clinic to really benefit aging people and people with degenerative disease.
I gotta say, I'm going to lower my moderator fee because the audience is just making it so easy for me. So this is just great. You guys have a lot of questions. So I'm going to throw the next one over to Ben. And Ben, that's the what's the link between the ion channel and pathology?
Well, I mean, that's a big question in the very early stages of understanding. It gets quickly very far beyond my technical capabilities. So as I said at the outset in my intro, I'm going to skate away from that kind of question and punt it over to Francesca and the balance of Team Pano that can respond to these in a much more acute way. I should probably stick to the business.
I'm going to give you another shot then. I'm going to give you a pass on. I'm going to give you a pass on that one. Admit that maybe you can check with Francesca and get back to on this question, which is an interesting question, right? So,
well, obviously very early in figuring all of this stuff out. So we're going to be working on that question for a long time, I would believe.
I figured that, yeah, so we'll follow up with the companies, and get some answers to all of these questions. I did actually expect that we get, given the somewhat, I don't want to suggest the similarity of approach, but basically you're under the same bit that Steve at Cohbar and Ben, you're under the same big tent in the mitochondrial approach, obviously different approaches, but within the same tent. So there are similar questions that have come in for both of you. And so, one question, I'm actually going to ask the first question, which actually comes from me, and then I'll actually ask the follow-up question, which may go a little deeper into the science, just as a warning, Ben, but we'll get there. So question is, why are you focused on mitochondrial dysfunction as such a central feature of aging and age-related diseases? That is the story of your company, right? So maybe just, Steve, let me have you have a shot at that, and then I'll turn it over to Ben.
Sure. You have hundreds of mitochondria in every cell. If they're not working properly, all sorts of things break down, from your immune system to your brain. Alzheimer's might be just a brownout. Why is that happening? As we know, as the mitochondria age in the body, we see more effects in the mutants occurring in the DNA, and the lack of repair of that over time leads to more heteroplasmy. And so you just have dysfunctional, or you're actually lacking mitochondria, each of which reduces the energy as well as the signaling that should be going on in the body. So this is underlying multiple diseases. And turn it around, when you think about what biotech and the pharmaceutical industry are focused on, and ask yourself, why have these chronic diseases been so hard to deal with, and the age-related diseases? And it's probably because we haven't been working at cellular level, that is the mitochondria.
Anyone comment on that?
Yeah, why don't I jump in to follow up? I'll take you to two or three sort of interesting perspectives. One is the unique spot of mitochondrial nuclear symbiosis in the genesis of biological complexity generally. I mean, we know that that's a very sort of fundamental movement. It's something that's completely unique in evolutionary history. 1.5 billion or so years ago, an aerobic alpha proteobacterium merged with an archaean cell, and all life changed forever from the 2 billion years that preceded it. So the common sense test suggests that there's a direct link between mitochondrial function and the homeostasis of biological complexity, which we might otherwise call resilience or health. I mean, this is, I think, something that we can sort of wrap our minds around generally, is that the mitochondria in this regard isn't just another organelle that arose through normal evolutionary processes. The other one, and I'll tell you this from the perspective of a middle-aged obsessive compulsive athlete, is that the mitochondria seems to be the sort of the central mediator of the non-pharmaceutical interventions that we contemplate to try and keep an optimum health. So if we take a look at some pretty interesting concepts like mitohormesis or mitochondrial allostasis, we can sort of dig into how the mitochondria behaves as a central mediator of the dynamic stresses that we have with our environment. So if we think of feeding and fasting, exercise and recovery, hot and cold exposure, hypoxia and hyperoxia training, et cetera, these seem to be mediated through the mitochondrial life cycle in a manner where, if we get it in the sweet spot of the biphasic dose response curve of these various stresses, the organism gets stronger and more resilient as the mitochondrial content and function within the individual cells in the organism gets stronger and more resilient. So we can see through the non-pharmaceutical lenses how that works when we're thinking about it from a conditioning perspective. I think the inverse equally would bear out if we're outside of that sweet spot for these various stresses. And I think the main one that the world is toiling with these days is just too much processed food that's laden with sugar, which in its own right would be a stressor that bears down on the mitochondriome and starts to crush it, and the organism falls apart behind it. So I think we can see it through that lens of the environmental relationship as well. The concept of mitochondrial allostasis, which I think Martin Picard kicks around quite quite a bunch. I think he's a really innovative scientist in this field, suggests that the stronger the mitochondrial function is within the organism, the more resilient the organism is to exogenous stresses. And then the third one I'd sort of look at is mitochondrial and life cycles, right? So we talked about the mitochondria having their own life cycle within the cell that is driven by its dynamic stress relationship with its environment. That's referred to as mitochondrial biogenesis. As that increases, it seems that the organism gets stronger. The mitochondria also control the cell life cycle as well as the organismal life cycle. So one of Doug Wallace's interesting observations in his 10th speech is that the only functional difference between your body 5 seconds before and 5 seconds after death is that your mitochondria stop respiring. And so we really understand, and looking at that edge case, so these various edge cases, that the mitochondria really sort of presents a very unique role in the functioning of complex biology. And
I'm gonna cut you off here, Ben, only because we're gonna move to the lightning round, and we got only a couple of minutes. And I got some really good questions. So and just a few. And Alex, I'm gonna let you finish. I have one question before I'm gonna direct it to you, which I think will relate really well with a lot of what we heard today. So this one's for Ashish, and this is the and I think that this question might be coming from an investor, so be careful. What's the extent of preclinical and scientific evidence that the Glyscend candidate is comparable to gastric bypass?
Yeah, I can disclose that if you sign an NDA.
So this is the lightning round. Do you want to just
yeah, so clearly the gastric bypass mechanisms are very complex, but what we have learned and found in our preclinical testing is along 3 dimensions. We have seen immediate glycemic improvement, we have seen weight loss, but more importantly, we have seen significant insulin suppression after postprandial spikes. And there are so combine that, these are the early signs that there's a gastric bypass or duodenal exclusion physiology working. Of course, more needs to be done, and we'll find out more when we go into clinic next year.
Great. Thank you so much, Ashish, for keeping that. And so we're gonna close out with you, Alex, in lightning round. It should be a straightforward question, fascinating question, which is, does in silico predict onset and or severity of age-related disease? Is it one or is it both? And how does that work?
So sure, actually very timely question. Today I gave a lecture at a very large conference organized by Intel in India with a lot of government officials, and it's many thousands of people, specifically on predicting severity of COVID patients and severity and the various risks. And we did it through in silico, and we did it in collaboration with Intel. And there are multiple ways of doing this, right? So multiple ways of skinning the cat, so to speak. And one way we employ it in silico, another way we employ at deep longevity. So in silico, we use generative biology, where we use those deep fakes, basically generative reinforcement learning, to use, and using age as a generation condition to extrapolate the patient into the future. So we train on millions, and then take one individual, basically ask the GAN to project that individual into the future with just one feature age, and GAN reconstructs the rest of the features, and we see what kind of risks the patient is facing. And we can do it with many data types. Another way of doing this is to use predicted biological age using multiple data types, right? You can even look at the person's face, and if they look significantly older, and you using just basic facial data, you can already project it into the future. And if they are older than their chronological age, most likely they are going to have higher hazard ratio. But we do it with blood tests, we do it with transcriptomes, with proteomes. And if you're anyone on this panel, if you're running a clinical study or preparing to run a clinical study, especially phase 2, or anything clinical, give me a call. I'll give you a bunch of biomarkers that will be basically fitting your budget, either cheap or free, and we'll be able to to do an aging clock augmented clinical study. I was very upset to see that, Restro Bio, for example, didn't do that. Amazing company, love the founder, but for some strange reason, big pharma thinking, it would be really nice to have a hematological aging clock predicting severity, mortality, but and risks, but also predicting biological age at enrollment, and also as you are using the intervention. Actually, for Juvena Bio, would be quite interesting to use our muscle transcriptomic aging clock, which we are using currently as workhorse for multiple applications. We patented that one too, granted. So very, very happy to collaborate.
I see a lot of follow-up here. So I'm gonna, since we are just out of time, we actually were out of time. First of all, I want to thank all the, there's still other questions here, quite a few. So I will get those to the organizers. They'll have them already, and we'll follow up directly with the panelists. So without further ado, I want to thank all the panelists for mostly adhering to those time limits, and actually thank the audience for great questions. There's a lot of people.
Yeah, you are the best. You are terrific. What a great panel. And I wish we did have another hour.